Hi, everyone. In this podcast, we're going to talk all about viruses and bacteria. Let's get started. I know viruses make us sick, but their simplicity and what they're capable of has always kind of been stunning to me. Viruses are so simple. All they have is nucleic acid. This could be DNA, but some viruses even have RNA instead of DNA. They then have a protective protein coat. We call that our capsid. Now, not all viruses, but some viruses, when they leave a cell or they lyse a cell, they'll take a little bit of our lipid membrane and use it as an envelope. And this will actually be their key to get into the next cell. Kind of tricky, huh? Viruses have two very different kinds of reproductive cycles. The first one that we're going to talk about is the lytic cycle. And that word lytic is referencing the fact that the virus will lyse a cell. And remember, lyse means to explode or pop open. So here's what happens. In this example, we're actually looking at a bacteria cell and a phage. And a phage is just a virus that infects a bacteria cell. So the virus lands on the bacteria cell, and its DNA is injected into the bacteria cell. This protein capsid is then just going to fall away. It served its purpose by delivering the DNA to the cell. The viral DNA will then mimic that bacterial chromosome by making a circle, and then it's going to hijack the cell. And what's going to happen is it may even just break apart the bacterial DNA, but it's going to make that cell make copies of the virus. It will make the genetic copies of the viral DNA and then copy that protein capsid, assemble the viruses, and this is going to keep happening until the cell gets so full of viral particles that it lyses or bursts open. And once that happens, baby viruses are going to be then spread throughout to go ahead and infect the next cell. And this is the lytic form of viral reproduction. Lytic viruses are viruses that will make you sick pretty quick after infection. Ebola, the common cold, these are lytic viruses. But what about viruses that don't make you sick right away, that get into your cells and kind of hunker down? We call this the lysogenic viruses. Okay, here's what happens. It starts just like the lytic. And again, in this example, we're looking at a bacteria cell and a phage. So the phage or the virus lands on top of the cell, injects its DNA, and the DNA enters the cell. Now, in lysogenic, through genetic recombination, the viral DNA gets incorporated into the host DNA. Once the viral DNA gets incorporated, we call that viral DNA prophage. Okay, and then those viral DNA genes are shut down, and it just quietly sits in the cell. The brilliance in this is every time this cell replicates, it replicates the viral DNA, and it's just going to keep replicating that viral DNA. So we'll go from one cell to maybe thousands to maybe millions of cells with that viral DNA. Now, in humans, this happens as well, and we don't really understand the cue, but there's some kind of cue that the viral DNA gets. It's usually accompanied with a stress, which is beneficial to the virus because then it knows it can kind of easily overwhelm, not easily, but could overwhelm an immune system. But basically, the lysogenic virus will suddenly become lytic. The viral DNA will activate and it will take over its host cell and behave just like a lytic virus would. Having that cell make copies of its DNA, have that cell make copies of its protein capsid, assemble the virus until eventually that cell could even lyse. Eventually then, if the organism's still alive, the virus will then go back into the DNA, becoming a prophage again, and then kind of resume that lysogenic form. Herpes is a good example of this. In humans, that's a virus that can do this. That's why someone who's infected with herpes may or may not be exhibiting symptoms because the virus might be in lysogenic and not lytic cycle. Here's something kind of cool. Not all viruses have DNA. Some viruses have the nucleic acid of RNA surrounded by that protein coat. Examples of this would be like the common cold, measles, mumps, West Nile, Ebola, polio. These generally have pretty high mutation rates going from RNA. So sometimes these are a little bit harder to make vaccines because of that. And that's why we have no vaccine for the common cold. We do have a really good vaccine for polio, and a brand new Ebola vaccine was just recently introduced. Some RNA viruses are what we call retroviruses. That retro kind of meaning backwards. So why are they called retroviruses? Well, they have RNA instead of DNA as their nucleic acid. And when the RNA is injected into a cell, an enzyme called reverse transcriptase actually takes that viral RNA. And I should mention reverse transcriptase does come from the virus. It sends it in with its RNA, and it takes that viral RNA and it copies it into DNA. Now, reverse transcriptase is not like DNA polymerase. It does not double check its work, so it messes up a lot. And therefore, there's a lot of mutations. This is an example of like HIV, which is why researchers are really struggling to make a vaccine, because the mutation rate is so incredibly high in HIV, and that's because of reverse transcriptase. Now, we have taken reverse transcriptase and we've used it in the laboratory so that we can actually take a human mRNA molecule, so it'll have no introns, just exons, and using reverse transcriptase, we can make that into DNA, and we can use that for gene therapies. As you know, viruses are considered non-living, but bacteria is a living organism. It's prokaryotic. Unlike you're eukaryotic, so it does not have any membrane-bound organelles or a membrane-bound nucleus. In a bacteria cell, it does have a chromosome in the nucleoid region, and the chromosome is large and circular in shape. Not only does bacteria have its large chromosome, that circular chromosome of DNA, it also has what are called plasmids. And plasmids are small circular pieces of DNA that can self-replicate. We're going to talk a lot about plasmids when we talk about DNA technology. I'm sure you guys have heard about antibiotic resistance. It's definitely a dangerous issue that we have to consider. So what does that mean? It means that within a bacteria cell, they have a gene that allows them to be resistant to an antibiotic. It allows them to survive that antibiotic. And here's what happens. So if you have the one cell that's resistant to that antibiotic, and you're taking an antibiotic, you're eliminating all this guy's competition, allowing it to continue to grow exponentially. Now you have a whole bunch of cells, bacterial cells that are resistant. And we're going to talk about in just a minute here how bacteria can actually pass genes off to another bacterial cell. So they can even sometimes share their antibiotic resistance. That's not good. Bacteria reproduces by mitosis, so essentially cloning itself. And you know the major disadvantage in that is a lack of variation. But bacteria actually has an answer to that. It can transfer DNA to other bacteria cells, and it has 3 ways of doing this. So let's talk about those 3 ways. The first one we're going to talk about is transformation. Transformation, we did this in the Piglo lab. So what happens in transformation is a bacteria cell will kind of randomly bump into a plasmid in its environment. Why would a plasmid just be hanging around in its environment? If another bacteria cell died, it may have like spilled all of its guts kind of into the surrounding area, and then this cell would run into it. And what the bacteria cell will do is it will just pick up that plasmid and have it within itself. And remember, once the plasmid's in the cell, it is self-replicating, so it can make more of itself. So that's our first kind of way bacteria can transfer DNA. And that's again called transformation. The second way bacteria can transfer information is transduction. And for this, we're going to need a virus. So remember, phages are viruses that infect bacteria cells. And what happens is the viral DNA will enter the cell, and it can cut up all of this bacterial DNA. And then the viral DNA will take over. It will use the bacteria cell's machinery to make copies of itself, make copies of its DNA, make copies of its protein capsid, all that good stuff. Well, what if, and this happens quite a bit, on accident, the protein capsids formed around the bacterial DNA instead of the viral DNA. This, phage, using air quotes, will go off and infect a cell, inserting bacterial DNA into a brand new bacteria cell. Then through genetic recombination, we will get a bacteria cell with genes that's from a different cell, essentially creating a brand new combination of genes. So that's the second way bacteria can transfer DNA. The last way bacteria can transfer DNA is called conjugation. And this is basically bacteria sex. So this bacteria cell right here, it has a plasmid, and that plasmid has a very specific gene that allows it to form a pilus. And a pilus is like a straw. And what it will do is this pilus will form like a bridge of cytoplasm with another bacteria cell. And then this plasmid will get copied. And as it gets copied, it's going to get fed into this second bacteria cell. So this bacteria cell will end up with a brand new plasmid it didn't have before. And this plasmid will remain in its cell. Now, this bacteria cell that got the new plasmid, it also can pass on this plasmid because it has that gene that allows it to make the pilus, which is kind of like that cytoplasmic bridge or straw, that can go ahead and find another cell and pass that plasmid on. Once a bacteria cell gets brand new DNA, it can actually get incorporated into its chromosome. And it does that just through recombinant technology. Basically, the DNA kind of gets degraded and re-pieced together, and we have a brand new recombinant chromosome. So this is part of why, evolutionarily speaking, bacteria has been so successful. It's not just cloned. It can actually create unique genetic combinations, which could help encourage it to survive. How do bacteria cells regulate which genes are on and off, and why do they? Well, the answer to this is operons. When bacteria cells copy their DNA into an RNA, specifically an mRNA molecule, it's automatically fed into a ribosome to be made into a polypeptide. Let's say you're an E. coli bacteria cell in somebody's gut. You have several genes to help break down and digest whatever that organism's eating so you can eat too. But what a complete waste of energy to be making different enzymes to break down different substances that may or may not be there. The answer is operons. They allow the bacteria cell to only make mRNA that will only make enzymes to break down what's present in order for them to consume that food. So let's meet the main players of an operon. An operon is basically a cluster of genes within the bacterial chromosome. One portion of the operon is a promoter. A promoter is DNA, and it's where RNA polymerase will attach to begin transcription. An operator is also DNA, and it kind of will act like a switch as to whether or not RNA polymerase can attach and go ahead and transcribe the genes. The repressor is actually a protein. It is not DNA. There is a gene that codes for the repressor, and it's usually upstream of the operon. We call this one of the regulatory genes. Let's go ahead and see how all of these guys work together to control gene expression in bacteria. Okay, let's take a look at how this works. Our promoter, remember, is where our RNA polymerase, that enzyme that is going to transcribe DNA into RNA, attaches to the promoter. And then what it will do, will go ahead and copy the DNA into RNA. The operator, remember, is the on-off switch. So how does that work? Well, this big red guy right here is my repressor. When it's attached to the operator, it acts like a physical block to polymerase. Polymerase goes ahead and attaches the promoter, and it's like, boom, you can't go here. And RNA polymerase falls off. So it's physically blocking transcription from occurring. We call this an inducible operon. And a good example of this is the lac operon. And I'll explain what the lac operon is in a little bit more detail in just a second. But it's inducible because the repressor is actually created in the form to attach directly to this operator and keep the gene off, okay? And this repressor is only going to come off that operator when lactose is present. So that's an inducible operon. A repressible operon is like TRP. And this protein right here, the repressor, when it's copied, it's just in the off position. So it's not attached to the gene. And then this gene can just get transcribed, at will. It's not until tryptophan shows up that this will attach. So it's a repressible operon. Okay, let's go ahead and take a closer look at the lac operon. All right, so E. coli lives in your gut, and E. coli likes to eat sugar. And one of the sugars it might eat is lactose, and it can't do that without the enzyme lactase. So lactase is the enzyme that will break apart lactose for the bacteria cell to consume. Okay, but why make lactase if there's no lactose around? All right, so here's what happens. All right, so here's my promoter, okay, right here. RNA polymerase is going to attach to that promoter, and it will go ahead and copy these genes in order to make lactase that will break down lactose. Right now, you haven't had any milk, so there's no reason for the bacteria to make lactase. So this repressor is attached to the operator. So the gene is off, okay, and the repressor is active. And it's actually transcribed to be active. As soon as it's made, it's active. It will attach to the operator, and it's going to stop transcription. All right. So now you drink a huge glass of milk, okay? And what will happen here is the lactose will actually bond to this repressor. It's a protein that's going to cause it to change shape. And when it does, like if you look down here, it bonds this, changes shape, it actually falls off the operator. So now what happens is RNA polymerase attaches and it can transcribe this gene. And once it does, it's going to go ahead and make that lactase, which will break down the lactose. All right, so how do I turn this back off? Well, I'm going to digest all the lactose, including this one, and then that repressor will go ahead and reattach to the operator, turning the gene off. The tryptophan operator is a little bit different. So the tryptophan, operon, is how the bacteria cell makes tryptophan, an amino acid. But if there's a whole bunch of tryptophan in the environment, why make it if you can just use it? All right, but this repressor is actually made in the inactive form. All right, so it's inactive repressor. All right, and so the gene is on, and I have an inactive repressor. All right, so what does that mean? RNA polymerase attaches to the promoter. There's no repressor on the operator, so RNA polymerase is free to go ahead and transcribe this DNA into RNA, which will then go on to make tryptophan. You sit down, you eat a whole bunch of turkey. That turkey has tryptophan. That tryptophan's in the bacteria cell, so why should it make it? What happens then is the tryptophan will then bind to the repressor, causing the repressor to change shape. This repressor will then attach to the operator and act as that physical block to RNA polymerase. RNA polymerase cannot transcribe the gene, and it cannot make RNA, stopping tryptophan from being made because it's going to go ahead and use the tryptophan already in the environment. This is an example of a repressible operon. All right, you guys, that's our podcast on bacteria and viruses.